Hello students, in this particular video we are going to discuss about all the spots which are going to come in your Biology Board Practical Examination 2024-25. So before we start with the spotting part, let me just give you an overview of the entire practical. Your practical would be for 3-hour duration and you will be scoring 30 marks in case you attempt the entire practical correctly. One major experiment that is going to carry 5 marks. This is going to be DNA isolation. One minor experiment, either it is going to be population density or population frequency. One of you, only one of the two, the teacher is going to ask you to do. Slide preparation 1 and 4, that means either you will be making the onion root tip slide, that means mitosis slide, or you will be asked to make a pollen germination slide, one of the two. Spotting, it is going to carry 7 marks, practical record for 4 marks and viva based on it, investigatory project plus viva based on it is going to carry 5 marks, so total 30 marks. Okay, so these are the list of the experiments. One compulsory experiment, this one is one compulsory experiment, DNA isolation. On my channel you will find all the videos related to your practical. From 1 and 4 you will be asked to make one. From 2 and 3 you will be asked to do only one experiment. Okay, so these are the list of the different spots. Okay, now how will you get the spotting? Total 7 marks are for spotting. 7 marks. Out of these 7 marks you are going to get 1 mark for each spot. For example, let's say flower pollination by different agencies. Either you are going to get wind pollinated flower or insect pollinated flower or bird pollinated flower. Only one of them you will get. Okay, let's say for example your teacher gave you a flower or an image or a photograph of a flower which gets pollinated by insect. Then pollen germination on stigma, then TS of testis or ovary. Let's say your teacher gave you this TS of testis. Okay, she's not going to give you both of them. Only one of these two you will get. Meiosis, any one of the stage the teacher can give you. Let's say your teacher has given you anaphase stage, anaphase 1 of meiosis. So let's say this is your third spot. Then blastula is there. Mendelian inheritance using seeds of different color or sizes, pedigree analysis. Let's say your teacher from here, she gives you this particular pedigree chart of blood groups. Let's say this is the fourth spot. Emasculation, tagging, bagging. You will not get all three of them, but you will get only one of them. Let's say your teacher gives you the image of a flower which is bagged. So you have to identify that the bagging has been shown here. Then Ascaris, Entamoeba, Plasmodium or any fungus which causes the disease. Out of all these four you will get only one. Let's say your teacher gives you the slide of Plasmodium. Okay, then symbolic association in root nodules, leguminous plants, cuscuta, lichens. Let's say your teacher gives you lichens. Okay, or flashcards showing homologous and analogous organs. Okay, you can get either any homologous organ from plant or animal or any analogous organ from plant or animal. You will get only one of it. Okay, so this way you will be getting 7 spots. 1, 2, 3, 4, 5, 6, 7. This is just an example I am telling you. This way the teacher will give you any of the 7 spots. Okay, so let's start with the first spot, that is pollination, a flower which gets a plant which gets pollinated by wind. So the plants are generally monoecious and bears unisexual flower. For this, what the teacher will do, she is going to show you an actual plant or she is going to give you an image of this kind of a plant. You have to identify that this plant gets pollinated by wind. Okay, so here you can see the male flowers, and in the spotting you will also have to write the comments and draw the labeled diagram. Please remember, comments plus labeled diagram, and you will get 1 minute 2 minutes for completing this entire thing. Okay, so these plants are monoecious and bears unisexual flowers. The flowers are small, inconspicuous, colorless, not showy, odorless and nectarless. Why they are supposed to be like this? Because they need not to attract the insects. This question could be asked in your viva also. Both stigma and anthers, they hang outside the petals, that is, they are well exposed. Pollen grains are light, small, dry, smooth, non-sticky to avoid clumping, and are produced in large numbers as they may be lost in transit. Stigma is long, sticky and feathery to catch floating pollen in air. And these are the adaptations that are meant for pollination by wind. So you are going to write in the end, on the basis of the above adaptations, we come to a conclusion that this image which has been provided to us is a plant which gets pollinated by wind. Okay, this is the way we do spotting. First we identify the characteristics and then we write on the basis of the above characteristics we identify the given spot as this. Okay, this is applicable for all the spots. Okay, next the teacher will give you this kind of an image. You have to identify that these are the flowers which are brightly colored for attracting pollinated insects. Flowers secrete nectar to feed visiting insects. Flowers have the landing platform for insects. Pollens are sticky, large, heavy and rough so that they stick to the body of insects. The stigma are also sticky so that the pollen deposited are not dispersed. If not conspicuous, then the flowers they form the inflorescence, and on the basis of the above characteristic we identify that this particular flower gets pollinated by insects. Next, pollination by birds. Flowers are large, they are strong because they have to bear the weight of the bird. So they are strong with abundant nectar or edible part, or they are odorless and brightly colored. Flowers are tubular and are curved that facilitate nectar sucking by birds. While sucking the nectar, the pollen gets deposited on their beaks and neck and are transferred to flowers of the plants they visit next. Next, this particular slide ma'am will show you. Okay, so you have to identify that this is pollen germination which is taking place on stigma. And you have to draw this diagram in which you will show the pollen tube, the antipodal cells, polar nuclei, egg cells, synergids. You will also show the male gametes traveling, two male gametes traveling you will show. Okay, and you will label this as the pollen grain. Okay, and you will show that you will write that this pollen grain is getting germinated here. So what are the characteristics that you are going to write? When the right type of pollen falls on the stigma, it germinates by absorbing water and nutrients from the stigmatic tissue. Pollen tube arises through the germ pore. Pollen tube grows in intercellular space of stigmatic tissue. Each pollen grain of flowering plant has two cells, vegetative cell and generative cell. The vegetative cell gives rise to pollen tube. The generative cell also descends into pollen tube and divides into two male gametes. The male gametes fuse with the polar nuclei and the egg cell. Next, TS of testis. So you will get this kind of a slide under the which will be placed under the microscope. You have to identify that this is the TS of testis. You're going to draw this kind of a diagram in which you will show these seminiferous tubules, then spermatogonia cells, then primary spermatocytes, secondary spermatocyte cells, spermatids and the spermatozoa. And you are also going to show the Sertoli cells and here interstitials or the Leydig cells. Okay, so these are the Leydig cells, spermatids, sperm, primary spermatocytes, spermatogonia, basement membrane, spermatocytes which are undergoing meiosis, Sertoli cells, okay, Leydig cells and blood vessels. These are the different labels that you have to show when you are drawing the diagram. So testis is surrounded by peritoneum and tunica albuginea. Testis is composed of numerous seminiferous tubules. Interstitial tissue contains the Leydig cell secreting the male sex hormone which is the testosterone. Each seminiferous tubule is surrounded by a basement membrane lined by a germinal epithelium. The germinal epithelium consists of two types of cells, namely the spermatogonia cells and the Sertoli cells. Okay, these are the spermatogonia cells here, and this is the Sertoli cell. Okay, so You have studied this in your theory also. Spermatogenia gives rise to sperm, and what is the function of Sertoli cells? They are large, tall, columnar cells which nourish the spermatozoa and secrete the hormone called as inhibin. Next, if you are going to get this kind of a diagram or this kind of a slide which would be placed under the microscope, you are going to identify it as the LS of ovary. You're going to draw this kind of a diagram in which you will show the primary follicle, primary oocyte inside the primary follicle, granulosa cells surrounding the oocyte, then cumulus oophorus. Here you will to show zona pellucida, theca interna. This is the follicular antrum which is filled with fluid. Here you are going to show ovulation taking place and the secondary oocyte being released, corona radiata, the layer around the secondary oocyte, then corpus luteum, the leftover part which is there after the release of the ovum of the Graafian follicle. This leftover part is called as the corpus luteum, and then it turns into the corpus albicans, and this layer is the germinal epithelium. It is covered by cubical germinal epithelium and tunica albuginea. Ovary contains outer dense layer, cortex, and inner loose layer called as medulla. Cortex contains many small and large spherical ovarian follicles containing primary, secondary oocytes. A full matured follicle is called as the Graafian follicle with a large central follicle cavity or antrum. After ovulation, a ruptured follicle transforms into a yellowish solid mass of cells called the corpus luteum, which secretes the hormone called as progesterone. Then you will get one of the slide of meiosis. Okay, so when the slide would be placed under the microscope, you will be clearly able to see which stage ma'am wants you to identify. In case the slide is not placed, then you will get an image. You have to identify by looking at it that which stage of meiosis is being shown to you. Okay, so let's say this is the image that has been shown to you. So you have to write draw the diagram and write the cell wall, nuclear membrane, nucleolus, chromosome, chromosome network showing distinct chromomeres, and this is the leptotin stage of meiosis 1. So chromatin fibers condense and form thick structures called chromosomes and appear clearly as long, thin and uncoiled threads. Sister chromatids are tightly bound to each other. Nuclear membrane and nucleolus are distinct. Centrioles are absent. This is the zygotine stage been shown to you with the help of this image. You have to draw the diagram and write cell wall, nuclear membrane, nucleolus, pair of homologous chromosomes showing synapses. So basically synapses is the main characteristic of this particular stage, and cell membrane. The chromosomes become shorter and thicker. The homologous chromosomes forms pairs called bivalent. This pairing is called as synapses. When the bivalents they align themselves opposite to each other, okay, so they form the bivalent and this pairing is called as synapses. Chromatids are not visible. A fibrillar ladder-like organelle called synaptonemal complex develops between the synapsed homologous chromosomes this way. The individual of a pair are similar in length and in position of their centromere. Pectine stage, this is the cell wall, nuclear membrane, pair of sister chromatids, centromere, nucleolus, non-sister chromatids of homologous chromosomes showing crossing over. This is the main characteristics of this particular stage, crossing over. So the synapse chromosomes continue to become short and thick. Chromatids of each synapse chromosome slightly separate and become visible. Two chromatids of chromosome are called as dyad. A group of four homologous chromatids or two dyads is called as a tetrad. Two chromatids of same chromosome are called sister chromatids, and those of two homologous chromosomes, that is the bivalents, they are termed as non-sister chromatids. For example, let's say these are the non-sister chromatids. Okay, these are the non-sister chromatids and these are the sister chromatids. Crossing over takes place, exchange of equal, what is crossing over? It is the exchange of equal segments of non-sister chromatids belonging to homologous chromosomes. Pectine chromosomes are long. This stage is considered more suitable for the study of chromosome morphology. Diplotin, cell wall, you can see cell wall, nuclear membrane, nucleolus, centromere, chiasma or the chiasmata and cell membrane. So this is the important characteristic of this particular stage. This cross-like structure which forms after the crossing over has taken place, this is referred to as chiasmata. So the two chromosomes of each bivalent move away and homologous chromosomes are held together at one or more points called chiasmata. The homologous chromosomes start separating. This is called as disjunction. However, the bivalents remain associated at chiasmata. The chiasmata mark the site where crossing over occurred during pectine. This question could be asked in your viva. Okay, chiasmata, it begins to be displaced along the length of chromosome and finally slips off at the ends of chromosomes. The nucleolus and the nuclear membrane, they begin to disappear towards the end of diplotin. Then comes the dikinesis stage, cell wall, nuclear membrane, homologous chromosomes showing chiasma and terminalization and cell membrane. So basically what happens in the dikinesis stage, the terminalization of the chiasmata takes place. The cross-like structure that was formed after crossing over, it terminalizes. It means that it comes to the tip and finally the chromosomes, they get separated from each other. So the homologous chromosomes, they appear thicker, nucleolus and the nuclear membrane disappear and spindle begins to form. Chromosomes become more contracted. Bivalents are more evenly distributed in the nucleus and migrate towards the periphery. Homologous chromosomes remain in contact with each other by terminal chiasmata. Okay, so basically we have studied about these stages. I'll write here. Meiosis, it's divided into meiosis 1 and meiosis 2. Meiosis 1 is further divided into prophase, metaphase, anaphase, telophase 1, 1, 1, 1 and meiosis 2 into prophase, metaphase, anaphase, telophase 2, 2. Okay, these are the second stages. Now prophase 1 is further divided into 5 substages which we have just studied, which all L, Z, P, D, D, leptotin, zygotine, pectine, diplotin, dikinesis. Okay. So we have studied all these. Now we are going to talk about this stage, metaphase 1. Okay. So you will be shown this kind of a diagram. You have to identify this is the cell wall, spindle fiber, homologous chromosome. They are arranged on the equator. This is the important characteristic of the cell which is in the metaphase stage. Whether it is the metaphase stage of plants or animals or it is the metaphase of meiosis or mitosis, this is the important characteristic that chromosomes, they align themselves along the equatorial plane. Spindle, cell membrane, pole. The bivalent homologous chromosomes, they arrange themselves at the equator of the spindle. Chromosomes become more condensed, spindle formation takes place. The spindle fiber gets attached to the homologous pair of chromosomes on their centromere with the help of kinetochores. The pairs move to the equator plates. Their movement is called congression. Anaphase 1, pole, cell wall, separation of homologous chromosomes due to contraction of spindle fibers, cell membrane. Two chromosomes of each bivalent move to opposite poles. Each pole has half the chromosomes with two chromatids each. The movement of chromosomes of a bivalent from equatorial plane to the pole takes place. That means the movement takes place this way from the equatorial plane towards the poles. From each tetrad, two chromatids of a chromosome move as a unit dyad to one pole of a spindle and remaining two chromatids of its homologue migrate to the opposite pole. This causes reduction of chromosome number. Each pole receives half of the chromosome of diploid number, hence the division is called reductional division. So half of the chromosome moves to this side, another half moves to this side and there is a reduction in the chromosome number which reach to the poles. So that is why this particular division is called as the reductional division. Telophase 1, cell wall, daughter nucleus with half the number of the parental chromosomes, chromatin, cell plate, nuclear membrane, nucleolus, cell membrane. The chromosomes, they reach the poles. Chromosomes at each pole uncoil and elongate. Nucleolus and the nuclear membrane reappear. Cytokinesis occurs to form two daughter cells and the spindle rays gradually disappear. Okay, now let's talk about prophase 2. This is the cell wall, nuclear membrane, nucleolus disappearing, cell plate, daughter nucleus with disappearing nuclear membrane and nucleolus cell membrane. So what exactly happens in this stage? Chromatids in a dyad are quite separate in the region of centromeres. Shortening and thickening of the chromatids takes place. Formation of the spindle fibers start. Chromosomes of daughter cells begin to condense and thick. Nuclear membrane and the nucleolus, they begin to disappear. Metaphase 2, cell wall, chromosomes arranged on equatorial plane. Chromosome, centromere, spindle fiber, pole, cell membrane. This way you are going to draw the diagram. The chromatids, they get arranged along the equatorial plane. Chromatids lie in center and their arms radiate towards the opposite poles. The centromeres divide into two and the two chromatids separate. Each chromosome is attached to the spindle by its centromere. Chromatids get arranged along the equatorial plane. Chromatids lie in the center and their arms radiate towards the opposite poles. Anaphase 2, cell wall, separated chromosomes after its division, cell membrane, chromosomes, they pulled to the poles due to contraction of the spindle fibers. The chromosomes moves towards the opposite poles due to contraction of spindle fibers. Centromeres now divide and chromatids are separated. The sister chromatids of each chromosome separate and migrate towards the opposite poles, being pulled by spindle fibers. Each pole thus receives haploid number of chromosomes. The chromosomes move towards the opposite poles due to contraction of the spindle fibers. Telophase 2, cell wall, nuclear, separation of the daughter cells due to cell plate formation. Nuclear membrane, chromatin network, cell membrane. So the haploid number of the chromosomes at the pole begin to uncoil and become thin and long. The nuclear envelope reappears and nucleolus are reconstituted. The group of chromosomes at each pole of the spindle gets enclosed by nuclear membrane. Nucleoli also reappear. The arms of chromatids get uncoiled. Astral rays and spindle also disappear. Cytokinesis occurs and four haploid daughter cells are formed from each parent. Okay, next the fifth spot is blastula. So you will be given a slide. You have to identify blastula and write the comment, draw the diagram and write the comments. This is the trophoderm or the trophoblast layer. This inner cell, this is the inner cell mass, and this is the albumin around the zona. So blastocyst is a structure formed in early embryogenesis of mammals which begins at day 5 after fertilization. Blastocyst comprises 70 to 100 cells. It is a thin-walled hollow structure and possesses an inner cell mass or embryoblast which forms the embryo. The outer layer of the cells is called the trophoblast which later forms the fetal placenta. The trophoblast surrounds the inner cell mass. A fluid-filled space enclosed by trophoblast forms blastocyst cavity or the blastocele. Okay, this is the sixth spotting, which is Mendelian inheritance using seeds of different color or sizes of any plant. For this, what is ma'am going to give you? She is going to give you yellow or green color beads. Okay, this particular practical is not practically possible by giving you the actual pea seeds. Why? Because these characteristics like yellowish seeds or wrinkled seeds, they are not visible because these days we get the hybrid peas. You must have seen that the peas that you get at your home, there is hardly any seed which is yellow in color or wrinkled in shape because hybrid varieties are available. So ma'am is going to give you yellow and green colored beads or wrinkled or and round beads so that you can easily distinguish between them. You can count them you will be able to count them and find out the ratio between these seeds. Okay, so and how are you going to write it? When the sample of the pea seeds are separated into round and wrinkled seeds or yellow or green seeds, put them in separate petri dishes or beakers, then they are found to be in the following ratio. So you are going to, on the basis of the seed shape, you're going to identify them as the round or the wrinkled shaped. You have to count the total number first, separate the round seeds and separate the wrinkled seeds. Okay. Then you're going to calculate the ratio. How will you calculate the ratio? 87 divided by 29. It comes out to be somewhere around 3. So the ratio is 3:1. Then you are going to write the inference which is written here. Okay, either this is going to be given to you or ma'am will give you some seeds which are going to be yellow in color or some seeds which are going to be green in color. So you will count, first of all you will count the total number of seeds, then you will separate the yellow seeds and the green seeds. You will calculate the ratio. How? 82 divided by 28, it comes out to be somewhere around 3. So the ratio is 3:1. What is the inference that would be drawn from this particular ratio? You're going to write the contrasting forms in both the traits of P, that is seed color and the seed shape, it show an approximate ratio of 3:1, which indicates or proves that the Mendelian law of segregation, which states that Mendel's law of which states that during gamete formation, the two copies of each gene in a parent separate or segregate, and each gamete receives only one allele. The result of the F2 generation gives the 3:1 ratio. Okay, seventh, spotting, rolling of tongue, pedigree chart. So ma'am is going to give you this kind of a chart. You have to, she can ask you the genotype of any of the individual. Okay, like how she'll ask you, she will say calculate the genotype, write the genotype of 2, 3. It means this is the first generation indicated by Roman 1, this is Roman 2, this is Roman numeral 3. So this is the first generation, second, and this is the third generation. So the third individual of the second generation is this one. So that means ma'am is asking you to find out the genotype of this particular individual. Okay, or what she can give you, she can ask you to write down the genotype of the entire pedigree chart. Okay, or she can also ask you what kind of the trait has been shown here with the help of this pedigree chart. So you are going to identify that this is autosomal dominant trait because the defective individual or the affected individual, it is present in the first, second as well as third generation. That means all the 3 generations. So you're going to identify that this is the autosomal dominant trait. Okay, now how are we going to write the genotype? See, non-roller female is indicated by a blank circle, roller female by a solid shaded circle, non-roller male blank square, and roller male by a shaded square. So non-rollers, they are, the genotype is small r, and rollers are the dominant ones. So their genotype is capital R or capital R small r. So there are two possibilities for the roller. This is the dominant characteristic. Okay. So how are we going to start it? In case we have to write the genotype of the entire pedigree chart, first of all, for who all are the non-rollers, the ones which are shown to you in the blank circles and squares. So there is no other option. Only small r is the option available to us for non-roller. So we are going to write small r for all the individuals which are non-rollers, which are shaded in, sorry, which are non-shaded. Here small r, this also small r, this also, and this also. Now because this individual is let's say this individual has 2 small r, so one would have come from the mother, one allele would have come from the mother, one would have come from the father. Okay, so that means the another allele which is available here is capital R, because in this condition only the person is affected. You can see here the roller, it is denoted by capital R, which is not possible here, the other option is capital R, small r. Okay, now we'll see the genotype of this individual. How? Small r, one small r would have come from here and one small r would have come from here from the father. The another possibility, the another allele which is possible here is capital R, because in this particular condition only the individual is going to be roller. Now here only these two are left. Okay, so one R has to come, one small r has to come from the mother, here also and here also. And the another possibility is because both these are rollers, so another possibility is capital R. They must have, they inherited the dominant allele from the father. So capital R, small r, capital R, small r. So this is how you are going to calculate the genotype of the entire pedigree chart. Let's move further. Widow's peak. This is another example of an autosomal dominant trait. How will we calculate? Dominant individuals are shown to you with capital W, and heterozygous condition also capital W, small w. In both these conditions, in both these genotypes, they denote the individuals which are showing the widow's peak. Widow's peak is the dominant character. It is shown by the solid shaded square or a shaded circle. Okay, so widow's peak female, widow's peak male, widow straight hairline female, and straight hairline male. They are shown to you by the blank squares and circles. Okay, so how will we start basically? So first of all, we are going to write this one, this option, recessive individuals. So wherever we find the blank circles or square, we are going to simply write small w, small w. Okay, now this small w in this particular individual, one allele would have come from the father and one would have come from the mother. Okay, so another possibility, another allele present here would be capital W, because in this particular condition only the father is going to show the dominant trait. Okay, here now these two individuals, they are going to carry one allele from the mother. That means one small w has to be there. Because both of them are showing the dominant trait, that means the another allele is capital W here. Okay, now let's see here, one small w, small w. Okay, this is small w, small w. This of this individual, one allele would have come from the father, one would have come from the mother. So that means the another allele possible here is capital W, because in this condition only the person is going to be affected, or we can say the person is going to show the dominant trait. So now the possibility of this individual is small w, because one allele has to come from here, and the because it is showing the dominant trait, so one capital W has to be there. Okay, now what is left now? This individual. Okay, यहाँ से let's start from here. This person would have inherited one allele from mother and one small w allele from father. That means the father would have had another allele as capital W. And here, therefore, this individual would have inherited one small w from here and one capital W from here. That is how we calculate the genotype of the entire pedigree charts. Let's move further. This is another autosomal recessive. Okay, this is the recessive trait, not the dominant trait. It is autosomal recessive trait. Okay, by looking at the pedigree chart, we can conclude this by seeing that there is skipping of generation. Skipping of generation. That means the characteristic is there in first generation, it's there in the second generation, and it's not there in the third generation. That means this is the recessive trait. Okay, so free earlobes, they are denoted by, this is the dominant characteristic at which is denoted by capital F and capital F small f, heterozygous and homozygous dominant. And attached earlobe, it's the recessive condition which is shown by small f, small f. So the individuals with attached earlobes, they are shown to you with the help of shaded squares and shaded circles. So let's first write this particular genotype. So here it's small f, 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 small f. Okay, this individual would have inherited one small f from here and one from here. That means the other possibility here of the allele here is capital F, because in this particular condition only the person has free earlobes. Okay, you have to remember this blank shows free earlobes. Okay, shaded shows attached earlobes. Okay, now here let's come here. The small f would have come from here, the small f would have come from here. The other possibility, other allele is capital F, capital F. Okay, in this particular individual, uh, it may have had the genotype capital F or capital F small f, because if this person would have inherited capital F from here, capital F from here, then only the person has free earlobes. If the individual would have inherited one capital F and one small f from here, then in this condition only the person would have had the free earlobes. Okay, now these two are left. So in this condition also capital F or capital F small f, this is the possibility. Here the possibility of the individual is capital F small f. It's not capital F here. Why? Because the parent, one of the parent is homozygous, another parent is heterozygous. So this individual would have inherited small f from the father and capital F from the mother. In this condition only the person will have the free earlobes. Now what is the possibility of the genotypes of these two individuals? So either they may inherit capital F from here and capital F from here, or capital F from here and small f from here. So both possibilities are there, and similarly here capital F or capital F small f. Next, ABO blood grouping. This is very easy. So you can be asked to write the genotype, or you may be asked to write the possibility that if this individual, let's say this individual marries an individual with blood group O, then what are the possibilities of the blood group of the offsprings. Okay, now let's write IA. There is only one possibility here, IA, IB, the two different alleles are there. Okay, so before I proceed, let me just tell you there are three genetic phenomenon associated with human blood grouping, that is multiple allelism, second is codominance, and third is dominance. Multiple allelism, why? Because there are three alleles which are responsible for determining the blood group. Why codominance? Because IA and IB, when both present together, then both are dominant. And why dominance? Because when capital IA and small I, they both are present together, then IA shows its effect. And this is the recessive allele, small I is the recessive allele. So you have to remember for your theory examination also that these are the three genetic phenomena associated with the human blood grouping. Okay, ABO blood grouping rather. So this is autosomal codominant trait when we are talking about IA and IB. Okay, so a person having AB blood group has this genotype, person having O blood group has this genotype, IA, IB, small I, small I, small I, small I, small I, small I. Okay, now let's move further now. This person would have inherited one small I from here and one small I from here. Because the blood group is A, the another allele possible here is IA. The another allele possible here is IB. Okay. Now this individual would have inherited one small I from here and one small I from father. That means the another allele possible here is IB. And similarly here, this person would have inherited one small I from father and IA from the mother. Okay. IA, small I, this particular individual would have inherited small I from here and IA from here. Now, another possibility of the allele present here is IA, small I. Now, this individual, it would have had IB from the mother and small I from the father. This individual, she must have had one IA allele from the father and one IA allele from the mother, or one IA allele from the mother and small I allele from the father. Similarly, this individual also, one IA or IA small I. So this way you are going to calculate the genotype. Next, color blindness. This is a little tricky. Okay, this is an X-linked recessive trait. Recessive because it's not been seen in all the four generation. It's a recessive trait and it is associated with the X chromosome. So this is the X-linked recessive trait. So here you will see that the individual which are normal, which have normal vision, the females, they are shown with the blank circles and they are represented by this genotype C plus C plus. The carrier female who has normal vision is denoted by C plus C and it represented by a blank circle. Again, female who is colorblind, affected individual, that is shown with a solid shaded circle and genotype is written as CC. The male who has the normal vision shown with a blank square and the genotype is C plus Y. Male who is colorblind, the genotype is written as CY and it is represented by a solid shaded square. You have to remember this. This will not be given to you. Okay, you have to remember this. Okay, now let's start writing the possibility. This female will start from here. The male who is colorblind, there is only one possibility for it, CY. So let's write the genotype here, CY, CY, CY. Okay. So here all the males are affected. Okay, so whenever you are shown, for your theory also you will remember, when you are shown a pedigree chart in which you see all the males affected and there is skipping of generation also, you will conclude that is the X-linked recessive trait. Okay, so this individual would have inherited Y from father, C from mother. Because this is normal, so the another allele possible here is C plus Y. Here you can see C plus Y, the male who has normal vision. Okay, C. Now this is also normal. One allele is C, that means the another allele is C plus. Now this particular individual, because he is a male, so one allele that he would inherit from the father is capital Y. Because he is normal, so he would inherit C plus from the mother. And here, because she is a female, the circle represents a female, so Y will not be inherited here. C plus would be inherited here. Okay, so this individual is going to carry C plus and it can have either this C plus or C from the mother. So there are two possibilities for this particular individual. Now let's proceed further. CY, this individual would have inherited Y from father, C from mother. He is normal, C plus. She is normal, C plus. Okay, here This is Y from father. This individual would have inherited Y from father, C from mother. He is normal, C plus. She is normal, C plus. Now this individual would have inherited Y from father, and because he is normal, he would have inherited C plus from mother. This female would have inherited C plus from the father, and either C plus or C from the mother, because in both these conditions the female is normal. What is left now? This. Okay, this is CY. This individual is normal because uh, this individual is normal, female, so one allele here is C plus. C plus C, this is the genotype of this individual. This individual would have inherited C plus from here and C from here. The another possibility for this individual is either C plus or C plus C. Here this male would have inherited Y from the father and either C plus, okay, only one possibility, not CY. This person's genotype, because he is a male and normal male, so the genotype is C plus Y. This individual, this female, she would have inherited C because she cannot have Y from father. She will have C from father and C plus from mother for being normal. So this is how you calculate the genotype. Okay, now let's move to the eighth spot, which is emasculation. So ma'am is going to give you either a flower in which she will cut these stamens, and she will ask you what is this particular process that has been shown to you, or she's going to show you an image, and you have to identify that in this particular image we can see that the stamens are being removed, emasculation is being done. And how will you write the comments? You are going to draw the diagram. You have to label that these are the stamens, these are the petals of the flower, this is the scissor, or a tweezer or forceps could also be used for this purpose for removal of the anther. And you're going to write the given image shows the setup of the emasculation. It is the step of artificial hybridization. Emasculation is the process of removal of the anthers before maturation of the stamens. It is done to prevent self-pollination. It is done with the help of forceps or scissors. In unisexual flowers, emasculation is not required. Okay, this kind of an image, it shows bagging. The given image shows the setup of bagging. It is the step in artificial hybridization technique. It is done on female flower. It is done after emasculation to avoid cross-pollination by undesired pollen grains. Rebagging is done after dusting desired pollen grains on the receptive stigma of the female flower. Tagging, okay, a tag been put over the emasculated flower. You have to identify that the tagging is shown here. So the given image shows the setup of tagging. It is a step in artificial hybridization. It is done after emasculation to write details about artificial hybridization experiment that is being carried out. The tag, it contains the following information: number referring to the field record, date of emasculation, date of crossing of pollination, name of the female plant, then cross sign and male parent, and finally the name of the breeder. Okay, the ninth spot is going to be any disease causing organism. So you have to identify, ma'am is going to show you the actual specimen of Ascaris or an image of Ascaris. You have to identify and draw the diagram. This is the female Ascaris: mouth, female gonopore, anus and tail, excretory pore, mouth bounded by lips, lateral line, cloacal aperture, pineal setae or spicules. The common parasite in small intestine of man, it is common in children than in adults. The body is elongated, unsegmented, cylindrical, pointed at both ends. The sexes are separate with marked sexual dimorphism. The male is smaller than female with curved tail. A pair of the curved pineal setae, that is the copulatory organs, and the cloaca is present. The mouth is terminal, triradiate, surrounded by three denticulate lips. The excretory pore is situated mid-ventrally, a little behind the mouth. Then this is the very most important characteristic. Ascaris causes ascariasis. You have to definitely write about the disease caused by the organism and its symptoms. So symptoms are abdominal discomfort, nausea, vomiting, diarrhea and colic pain. The toxin produced by Ascaris may interfere with the protein digestion. Then this kind of a slide or image will be shown to you. You have to identify that this is an amoeba histolytica, the ectoplasm, endoplasm, food vacuole, plasma lemma, nucleus, endosome and pseudopodium. They are going to be labeled in the on the diagram. It is a pathogenic intestinal monogenetic parasite occurring in colon of man and causes amoebic dysentery or amoebiasis. There are two forms of it: magna, which is pathogenic form found in the mucosa and submucosa of intestine, forming ulcers, and minuta, which is non-pathogenic form. It is found in the lumen of intestine. Entamoeba has no contractile vacuole like amoeba. The minuta form and cyst. A mature cyst is called quadrinucleate cyst with 4 nuclei and 2 chromatoid bodies. Quadrinucleate cyst is the infective stage, and infection is oral through contaminated food and water. Houseflies sitting on fecal matter of host containing cyst may transfer them to foodstuffs. Its adult is called as the trophozoite. It causes uh, the, uh, the disease which has the symptoms such as diarrhea with loose stools, dysentery with mucus, traces of blood, in severe cases liver and other organs may be affected. Plasmodium. This kind of a slide will be shown to you. You have to identify them as plasmodium. You have to draw the diagram. Okay, so you have studied this in your theory also. Malaria, when we have talked about malaria. So when the malarial parasite it enters through the in the body, when it enters in the body through the bite of the mosquito, then the sporozoites they are released. They reach the liver, then they divide there. They enter the RBCs. They rupture the RBCs. They form the gametocyte cells. Okay, they are picked up by the bite of another Anopheles mosquito, and there is fusion of the male and the female gametes forming the zygote, and further development takes place forming the sporozoa, which are further picked up by the Anopheles, female Anopheles mosquito. It is intracellular, pathogenic, blood parasite of man and other vertebrates. The life history is completed in two hosts, male and female Anopheles mosquito. Asexual cycle is passed in man by a process called schizogony in liver and RBCs, which is a type of multiple fission. Sexual cycle is completed in female Anopheles mosquito. When an infected female Anopheles bites man, sporozoites are introduced into blood. Then they reach liver cells through the bloodstream and multiply to form the merozoites. After few schizogonic cycles in the liver, the merocytes they enter the RBCs and feed on the contents of RBCs. Female Anopheles mosquito sucks the gametocytes with blood for further development. Symptoms of malaria, they cause malaria, and the symptoms of malaria is recurring fever and flu-like illness including shaking, chills, headache, muscle aches and tiredness, nausea, vomiting, diarrhea and anemia. Next, ringworm. Okay, this image you have to identify that this is a ringworm. Okay, one thing I forgot to mention about the name of the disease here. You have to write that it causes amoebiasis. Okay, you have to mention the name of the disease also. Here, Plasmodium, it causes malaria. Entamoeba histolytica, it causes amoebiasis. Ascaris, it causes ascariasis. Fourth one we are going to talk about now, ringworm. So ringworm is common fungal skin infection that comes from the microscopic organism called dermatophytes. Examples of dermatophytes are Trichophyton, Microsporum and Epidermophyton. These organisms survive by consuming the dead layers of the outer skin of human and animals. The ringworm infection can affect any part of the body, scalp, feet, groin area or the nails. Symptoms of ringworm: reddish, scaly patch or bump on the skin, itchy, uncomfortable, which eventually resembles a ring with bumpy borders. Patch in between the toes can cause skin to crack. On scalp, it starts like a pimple that gets larger and becomes patchy, flaky, and scaly. It often becomes tender or red and causes baldness in the scalp. Sometimes nails become yellow, patchy, and thick. Now, the 10th spot, symbiotic association in root nodules of leguminous plants. The root nodules, this kind of an image will be shown to you, or the actual specimen will be shown to you. You have to identify that these are the root nodules, and you will label, when you will draw the diagram, you will label this is the root of leguminous plant, these are the root nodules. Root nodules are commonly found in the roots of the leguminous plants. They are formed due to association with the nitrogen fixing bacteria Rhizobium. Rhizobium is gram-negative bacteria which forms a symbiotic relationship, which is mutualism, where both plants as well as the bacteria are benefited from each other. Root nodules contain pink color leghemoglobin pigment. It also contains enzyme nitrogenase, which helps in the formation of ammonia from atmospheric nitrogen that can be used by the leguminous plant for its growth and development. Bacteria receives nutrients and shelter from the plant. What is mutualism? You have to write this also. It's a relationship between two organisms in which both of them benefit from each other. Cuscuta on host, you have to identify this yellowish colored stem-like structure. They are cuscuta on the which are growing on the host plant. So cuscuta, which it is also called as the dodder or the amarbale, it is a parasitic plant, or it is ectoparasite that is commonly found growing on hedge plants. It has lost its chlorophyll and lives in the course of evolution. Why? Because it need not to have chlorophyll, it need not to make its food on its own, because it derives its food from the host plant. It derives its nutrition from the host plant, which is parasitizes, and it can kill the host plant. Stem is thin and slender shaped without chlorophyll. It fixes with host plant by a special structure called hostoria. Hostoria, it forms direct connection with the vascular bundle of host plant and withdraw water, carbohydrates and other solutes. Stem of Cuscuta winds around the stem of the host plant. It has no fully expanded form of leaves. Scale-like leaves are present that have no chlorophyll. Roots are temporary and die as soon as it makes the connection with the host plant. Then this kind of a relationship which is shown to you with the help of this image, it is parasitism. Then you're going to write two statements, simple statement about parasitism, that it is the relationship between two organisms in which one of them benefits and the other gets other does not get any benefit, other gets harmed. Okay. Lichens. Okay, so ma'am will show you the actual image of lichen or the diagram of lichen. You have to identify that these are the lichens, which are composite organism representing a symbiotic association, which is mutualism, in which both the organisms benefit from each other. So it's a relationship between fungus and the photosynthesizing algae or cyanobacteria. Algal component is called as phycobiont, whereas the fungal component is called as the mycobiont. The algae prepares the food for fungi, and fungi provides shelter and absorbs the minerals, nutrients and water to the algae. They grow on land, rocks, tree trunks and wall of houses. Three types of lichens are there: crustose, foliose and fruticose. Then you are going to define what actually mutualism is. It's a relationship between two organisms in which both of them benefit from each other. Next, the last 11th spot is homologous organs in plants. Then we are going to see homologous organs in animals, then analogous organs. So this is the 11th spot. So either ma'am is going to give you this image. You have to identify that these are the tendrils and these are the thorns. These are the thorns of pomegranate. Both of them are homologous because they have the same origin, but they have different functions, so they are homologous. So tendrils of passion fruit and thorns of pomegranate are structurally and functionally different, but they have same origin. That is, they arise from, both of them, they arise from the axillary bud. Then what is the function of tendrils? They help in gaining support or climbing, while the thorns, they help in protection. Okay, this you can see bougainvillea and cucurbita. So these are the thorns of bougainvillea. These are the tendrils of cucurbita. Both of them are arising from the similar place, so they have the same origin but they have the different function. They are therefore they are homologous. So these are the organs or parts which are quite dissimilar to each other in appearance and perform different functions, but they have the same origin and anatomy. This is the definition of the homologous organ. The differences in their function is the result of divergent evolution due to adaptive radiation to different habitat and ecological niche. So these organs are called as the homologous organs. For example, these two organisms, they have come from different areas. Okay, no, these two organisms, they have moved to different areas. They have started to live here. Okay, so their organs, they adapted themselves to according to the needs of different regions, but both of them, they have the same origin. So this shows the divergent evolution. Analogous organs in plants, you can see the tendrils here in this pea plant and tendrils here in grapevine. This is grape, this is pea plant. Both of them are tendrils, but they are arising from different areas, different regions. So these are structurally similar, functionally similar, but structurally they are different from each other when we talk about their origin. The stem tendrils and the leaf tendrils. The tendrils are analogous with one another, being structurally and functionally similar irrespective of their origin. Example, the tendrils of pea and tendrils of vitis. Tendrils of pea are modification of the leaf, and in vitis it is the modification of the terminal bud. Okay, here you can see, this is the another example. Okay, here you can see the thorns and these are the spines. Both of them, they serve the function of protection, but they have different origins. They are the modified leaves in cactus, but they are arising from the axillary or the terminal buds, these thorns. Okay, so thorns and spines, the thorns are and spines are analogous structures being defensive in function. Both the function of both the organs is defensive in nature. The thorns are modifications of the axillary or the terminal buds, and spines are modifications of leaves. Example, thorns of pomegranate and spines of prickly pear. In plants and animals, there are several organs or parts alike in their function and appearance, but different from each other in their origin and anatomical structure. These organs are called as the analogous organs. The similarity is result of convergent evolution. It is showing convergent evolution. Convergent, convergent, convergent evolution, which is adaptation to similar habitat and identical ecological niche. It means that there are two different organisms which have moved in a particular area, and they are going to adapt themselves according to the needs of these particular area. Therefore they are going to develop these structures which are going to help them to survive in this area. So this shows the convergent evolution. Homologous organs in animals. So these are the four limbs of these organisms: man, cheetah, whale, bat. They have similar origin but they have different functions. So wings of birds, four limbs of mammals, reptiles, frog, all have the same bony elements, that is humerus, radioulna, carpals, metacarpals and phalanges. This is humerus, radius ulna, carpals, metacarpals and phalanges, but perform different functions, flying in birds for holding or walking, etc. in other. There are organs or parts which are quite dissimilar to each other in appearance and perform different functions but have the similar origin and anatomy. This is the definition of homologous organs, which we have already discussed, and it is the result of divergent evolution. Last, analogous organs in animals: wings of a dragonfly and wings of a bird. Both the wings of different organism, they are helping in flying, but their structure is different, their origin is different, so therefore they are analogous organs. So analogous organs are basically in plants and animals, these are the several organs or parts alike in their function but and appearance, but different from each other in their origin and anatomical structure. These are the analogous organs. Oh, this is wrong, this is convergent. They represent convergent evolution. So wings of dragonfly, cockroach, butterfly and of birds, They are the analogous organs, mandible of cockroach and mandible of vertebrate. They are again analogous. Both of them are helping in chewing, but their structure is different. So with this we come to the end of all the spotting. So good luck for your board practical examination.